Hello and welcome to today's webinar, Brewing with Wild Things, a genetic survey of mixed culture fermentation in the U.S. I'm Ryan Gutierrez, Senior Product Marketing Manager at GENEWIZ, and I will be moderating today's session. Please join me in welcoming today's presenter, Dr. Daniel LePage. He is the Quality Manager at Creature Comforts Brewing Company in Athens, Georgia, where he oversees everything from raw ingredients to packaged product. But he especially has a passion for the microorganisms used in between. He earned his BS in biochemistry from the University of Tulsa and his Ph.D. in biological sciences from Vanderbilt University. If you have any questions for Dr. LePage during today's presentation, please enter them in the Q&A section, and we will try to address as many as we can before the end of the session. With that, please welcome Dr. Daniel LePage.
Hi everyone. So to start this afternoon, we're just going to have a little overview of the brewing process. So when a brewer is looking to generate beer, they're going to utilize water, grains, and hops to actually create what's just a sugary microbiological media called wort. Then we're going to take that wort, we're going to send it to a stainless steel fermenting vessel. And once it's there, we're going to add a defined strain of brewing yeast, let them do their thing for a couple weeks, and then we'll have our beer. Now, one of the really common sayings in the industry is that brewers don't actually make beer. Brewers make wort, yeast make beer. And so your choice of yeast is actually incredibly crucial for the beer that you might be brewing. When brewers are deciding which yeast that they're going to use, they're actually going to be choosing between just two different species the vast majority of the time. This is going to be Saccharomyces cerevisiae, your typical yeast, and Saccharomyces pastorianus. And this is really the only difference between ales and lagers. It has nothing to do with the ingredients or one being heavy, one being light or higher or lower alcohol. The only difference between an ale and a lager is simply what species of yeast the brewer decided to ferment that beer with. Now, out of these two species, there are hundreds of defined strains available. You can buy them from many different commercial companies. They will send them to you. They're very well classified. A lot of them, we have full genomes available, and we know the flavor and aroma profiles that they're going to generate, their preferred temperatures, their attenuation, all kinds of properties for these defined strains. But if you want to get a little bit wild with things, you can also use bacteria in conjunction with yeast while you're brewing. In most of these cases, brewers are going to be using either lactobacillus or pediococcus species, and these are specifically used to generate acid and really lactic acid. And this is something that we do at Creature Comforts. We use a technique called kettle souring, and we have a house strain of lactobacillus. And what we're doing there is before we send the wort to this fermenting vessel and add yeast, we're actually going to keep it in the brew house in the boil kettle. We'll add our lactobacillus. We'll let it to actually generate lactic acid and to start to sour that beer. And when it gets to just the right tartness that we want, we can simply turn on the boil kettle, sterilize the wort, then send it to the fermenter and add our yeast to make our beer. And using that technique, we can get these nice, lightly tart beers that are really refreshing and great during the summer. But that's not a technique that every brewery is using. The vast majority of them are sticking to just your typical brewing yeast. On the other hand, if you want to go completely off the rails, you can actually fall into the realm called mixed culture fermentation. And this is really just everything that doesn't use defined brewing strains. As you can imagine, this covers an untold number of techniques and processes and species that might be used in the beer. And whenever you're looking at mixed culture fermentation, it's really common to actually ferment the beer in wood for extended periods of time. So at Creature Comforts, we have large wooden fermenters. We also have old wine barrels that we can ferment our beer in. And instead of fermenting for just a couple weeks, it's really, really common for these to stay in these wooden vessels for months or even years. On top of that, you can also have what's called a spontaneous fermentation. And this is where you're simply going to expose the wort to whatever might be in the air, and it'll happen to fall in, and then inoculate the wort and then ferment your beer. To do this, you can take your wort, you can wheel it outside if you want, and just let whatever's outside float into it. Or you can have a dedicated room called a cool ship. And cool ships are typically lined with wood. You let the beer cool in the cool ship, and the microorganisms that are naturally present in that wood, in that room, will inoculate the beer and then begin to ferment it. You can also get some of these other organisms from ingredients if you add fruit or anything weird like that. And a lot of breweries actually have a house mixed culture. So this is a culture that they've used repeatedly over the years. They might curate it for flavor and aroma, but these could have incredibly diverse origins. They might have taken a swab from a brewer's beer, or they had some weird fruit that they really liked, or dregs from a bottle of beer that they tried in Belgium and thought was delicious. A whole variety of mixtures that can go into a brewery's house mixed culture. But this realm of brewing is a really quickly growing field in the US. There are over 7,000 craft breweries in the United States right now, and there are actually several of them that are solely doing mixed culture fermentations. They no longer use defined brewing strains because they really like the flavors and the profiles that they're able to get with these more varied fermentation profiles. So a really obvious question to ask when you're looking at these beers is what species are actually present? What is actually fermenting this beer? And this is an obvious question. We're not the first ones to ask it. There have been previous studies. These are just some of them. But a lot of these studies have largely focused on one brewery and maybe even one culture. And they might have looked at that culture over time, or they looked at it in different vessels or under different fermentation characteristics. But it's really just been one brewery, one culture. And they've also utilized 16S and ITS sequencing. And this is particularly tricky because a lot of the things that we see in brewing are very, very closely related. And so trying to get resolution for actual species identification is difficult with 16S and ITS sequencing. So for our project, we actually wanted to utilize shallow shotgun metagenomics. And by utilizing this, we wanted to actually get down to those defined species, especially the closely related ones. We also hope that this could allow us to see different strains that might be present in our beers. And it also allow us to look into various metabolic pathways and genes that might be present in these organisms used in mixed culture. For this, we're also interested in both early fermentation, because that's going to show us, what is responsible for a large amount of the sugar utilization and the flavor formation, and also later fermentation. If we take that same beer and we look at it a year after it's been sitting in a wine barrel, now it doesn't have much sugar left, but it's still maturing. It still has flavor and aroma compounds that are being generated and altered, and also curious about the product stability over the years. Is it eventually going to turn into vinegar, or are we going to have a tasty product if we open that same bottle a year from now? Previous studies have looked at this, and they've shown that in general, with mixed culture fermentations, that it tends to be fairly yeast dominant early on and can become more bacterial dominant later in the fermentation. Now, everyone knows drinking is always better with friends, and we knew that if we weren't the only ones going to be interested in this, we wanted to invite some of our friendly breweries. So in the end, this project included 18 participating breweries, and we were really humbled by everyone who agreed to join. All of them have agreed to have their anonymized data shared, so I will not be able to mention any of the other breweries that were part of this project, other than say Creature Comforts was one of the 18. And these breweries are all based in the United States, but they cover a vast gamut, small breweries, large breweries, East Coast, West Coast, and everything in between. For general study, philosophy, and design is quite interesting because for this project, full controls are practically impossible. For these samples, we're looking at different origins, different locations, different wort streams, handling techniques, different fermentation vessels and temperatures, different ages, and it's really not possible for us to control all of that across these different breweries. And so what we did was we really allowed each brewery to dictate their own interests. We told them what technology we were going to be using and what we planned to get from it, and we just asked them, what would you like to know about your particular mixed culture, about your techniques, or about the beer that you are fermenting? And with this, we hope to get a general snapshot of just mixed cultures across the US right now. How unique are these cultures? What's everyone working with? Can we see differences based on other people's techniques and things like that? And so in the end, we ended up with 37 different samples from these 18 breweries. And it we're really, really happy. It just covered the gamut. People send us a lot of crazy stuff that's been a lot of fun to play with. So some of these samples were fermented in what's called fooder, which is basically just a large wooden fermentation vessel. Some of them came from cool ships and were spontaneous fermentations. One sample in particular, they just took their wort, they wheeled it out into a vineyard, and they let whatever fell into it ferment it. We have some wild yeast. We have a lot of samples from barrels. We also sequenced our own house lactobacillus strain. We have a lot of house mixed cultures, cultures that these breweries have been using for years, in some cases decades. We have some samples from bottles, so we can look at product stability and maturation over time. And we also have some contaminated barrels that particular breweries were interested in. Why did this one barrel of beer fail our quality control checks, and what can we do in the future to stop that from happening? For the actual sequencing of these 37 samples, we used 5 lanes of paired-end Illumina high-seq sequencing. Many of these samples were quite difficult, as they've been sitting. A lot of them have been sitting in a barrel for up to a year, and so there weren't many cells left, and some of the DNA could be quite degraded. So some of these had DNA as low as 200 nanograms total. Only one sample ended up failing QC for not having enough DNA, though. And in general, we got fantastic library preparation. We had 53 to 88 million reads per sample, an average of 65 million reads with quite good quality scores. When it came to the data analysis, these first rounds, we've had 2 scientists working with us on the analysis pipeline. This is Henk Denbacher from the University of Georgia, as well as Andrew Brooks, who is a postdoc at Stanford University. And these 2 have really done a deep dive into the sequences. We ended up getting about 400 gigabytes of raw reads from this project. But all the data that I'm going to be sharing today actually came from Henk's analysis. For this, he formed the contigs using spades with metagenomic settings, and then bin them using metabat. One thing to take note of is that all the following graphs that I'm going to show you show the prokaryotic and eukaryotic reads together. Since the eukaryotic genomes are quite larger, this means these graphs are biased towards eukaryotes, but that's just something to keep in mind as we're going through these data. Get into it. Here we have some banked cultures from 3 different breweries. So these are cultures that they would take from their freezer, and then they would pitch them into a fresh batch and just let it ferment. And these are cultures that they've worked with for years, and something that is really a workhorse in their brewery. We can see that the sample from brewery 1 is dominated by yeast, in fact, Brettanomyces bruxellensis. And so by pitching this, they are going to get strong fermentation in the beginning. They're going to have a lot of yeast present in the sample. But what's interesting is that brewery 2 and brewery 3 samples are actually dominated by bacteria. And so they're going to get a lot of acid formation in the beginning. Brewery 2 has a lot of lactobacillus acetotolerance, and so they could get a large amount of acid in the beginning of their fermentation. Brewery 3 actually has a lot of acetobacter in it. And this is something that they might actually want to keep an eye on. If you have a lot of acetobacter, you could get a lot of acetic acid formation, and this could be eventually leading to off flavors in their beer. Some acetic acid can be acceptable in beer, but at a certain point, no one really wants to drink vinegar, and so this is something that they could look into to curate their house culture. We can also look at the same culture, early fermentation versus later fermentation. And so for this particular sample right here, this one brewery, and we see that the early fermentation is dominated by Brettanomyces bruxellensis. This is pretty expected given previous studies. And then later on in fermentation, we see that the bacteria start to come up. So what's interesting here is that we see Pediococcus damnosus actually increase a decent bit in this population if we just count by raw reads. And one organism that we don't even see in the early fermentation is this Brettanomyces custarsianus. It's quite prevalent later in fermentation here, and we didn't get a single contig from that in the early fermentation sample. We do the same thing for a different brewery. We can see that this one is actually bacterial dominated early on. It has a lot of lactobacillus, and what's interesting is that it actually loses this Lactobacillus hilgardi and amylovorus with time. Later on fermentation, we don't see those organisms anymore. So that's something that they might have to keep in mind when they're looking at, should they take a sample from early fermentation or later fermentation to pitch a new batch of beer? They might actually be losing organisms throughout these batches. Another interesting thing that we see with this sample is that we get the same arrival of Brettanomyces castricianus later on in fermentation. And so this is pointing to something that this might actually be a signature of this species. It might be incredibly slow growing, or it might just prefer the environment that our later fermentation beers have. The higher acidity, the alcohol content, no to little oxygen, things like that. And we also see a pickup of Pediococcus. Is this something that we just didn't see in the early fermentation sample? Is this something that is on the wood that it's fermenting in? Is it something that the brewery might be interested in keeping an eye on? We're not completely sure. Here we have a couple more late fermentation samples. And what's interesting here is we see that these are actually yeast dominant. So we would not expect that for these samples, considering they do contain bacteria. And so previous studies would tell us that eventually that bacteria should become dominant and the yeast should die off. But these keep a large amount of their yeast late into fermentation. Once again, we can't judge full ratios off of just these read counts, but in general, it looks like the yeast are very happy and healthy even in these later samples. One really fun set that we got to look at is a brewery that sent in 2 samples of what they considered both to be their house mixed culture. And even though they claim this as the same culture, these 2 samples actually only share one organism, the Lactobacillus brevis. And so this is really interesting that if they're using one of these to pitch new beers, they're probably going to get very different flavor profiles over time. And if they consider both of these their house mixed culture, that's obviously not correct. One of these is different. In fact, we see that one of the samples does contain a lot of bacteria that is very typical for spontaneous fermentations. So it's possible that they had a combined spontaneous and mixed culture fermentation, house mixed culture, that then later on got forgotten about, and it's still maintaining these bugs from the spontaneous fermentation. Some other things we can look at 2 different foeders, and these are those large wooden fermenting vessels. These are 2 foeders from the same brewery. They've been at that brewery for years. They've had multiple batches of beer go through them. And what's interesting here is that on a species level, these are actually very, very similar. Although one does contain Brettanomyces bruxellensis and Pediococcus pentasatius, that the other does not. So for this particular brewery, if they like the flavor of beer that they're getting from one foeder over the other, this could inform them on what cultures they might need to move from one foeder to the other, or what they need to eliminate from one of their foeders to generate a more even and consistent product. But in general, the species between these 2 are actually quite similar. We can also look at spontaneous fermentations, and these were all in barrels for right at a year. One sample was 11 months, another 12, and another 13 months. One thing to note here, we see an incredible diversity with these spontaneous fermentations. These are actually some of the most diverse samples that we saw. In general, each of these has 8 organisms. The most diverse sample that we see in our data set had 14. And these are nowhere similar to each other. We see a vast array of different organisms, vast array of whether they are more bacterial or yeast dominated. And so these spontaneous fermentations where you're purposely just letting whatever weird thing happens to your beer obviously seems to work because these are incredibly varied. And here we can see ones that are difference between 2 and 3 are very yeast dominant, but sample 1 is actually bacterial dominant. And what's really fun is this third sample right here has Hanseníaspora uvarum, which is a wild yeast that's very, very common on grape skins. And this is the beer that the brewery just wheeled it out into a vineyard. They let whatever was in the vineyard ferment their beer. And so no surprise, it actually has a grape yeast in there. We can also look at spontaneous fermentations in the bottles. Here we see some species that we actually don't see anywhere else. And for this particular one, it retains some of these strange organisms that we see in spontaneous beers all the way through the bottle, even though it's been in bottle for close to a year. And so this showing that these organisms that are common to spontaneous beers that are controlling the fermentation could also be controlling the maturation of the product and the long-term stability of the product, because they are able to stick around. This is quite fun. This is just showing our overall data, all the samples, all the diversity. And you can see that this is incredibly diverse. This isn't just one or two species of yeast. In fact, overall, from this project, we saw 56 bacterial species and 10 different yeast species. The most common ones that we found were actually Lactobacillus acetotolerans and Pediococcus damnosus, which we found in 27 of the 36 samples. And the most common yeast is Brettanomyces bruxellensis. It's not even a species of Saccharomyces. That was dominating most of our samples. So some of the initial conclusions from this very, very raw first round analysis is that bank cultures cannot really be considered gold standards. So these need to be checked, they need to be verified. That your house mixed culture sample might not actually be what you think it is. In fact, brewery that had two that they thought were the same were not. And it might be dominated by some bug that you don't actually want to pitch into fresh beer, such as an acetobacter or something like that. We see that Lactobacillus acetotolerans is incredibly common. And this should really inform brewery contamination checks for any brewery that's also doing clean fermentations. Because Lactobacillus acetotolerans in the brewing community is well known to be difficult to see with your typical contamination checks. And if this is something that's really, really common in your culture, though, you need to make sure that your processes are good enough to see that and to catch it in case it might be infecting your other beers. We also see that despite previous results, cultures can be dominated by bacteria and yeast later on. And that cultures can be stable within a brewery, and unique techniques do show up in the represented species in the final product in bottles. Of course, this isn't just the only analysis that we want to do with this data. One of the great things about metagenomics is it gives you an absolute pile of reads to play with. And so some of the things that we're going to be doing in the coming year are we're actually going to be looking not just at species ID, but we're going to be looking for unique strains within all of these samples. We're also looking to analyze the close to 900,000 identified genes that we got with these reads. And with that, we want to look at metabolic pathways, specifically sugar utilization, acid production. We also want to look at novel hop resistance genes. So there are several hop resistance genes similar to drug resistance genes known in the brewing industry that allow microorganisms to survive in a hoppy environment, which are naturally antibiotic and antimicrobial. And so if we can find any of these new hop resistance genes, that'd be quite interesting. We also want to look at novel diastaticus genes. And diastaticus genes are simply genes that allow yeast to ferment sugars that they usually wouldn't be able to. There are at least 3 known right now, STA1, STA2, STA3. We would love to find STA4 in this data set. We also want to look at links to flavor characteristics. These breweries have incredibly diverse products, and some of them have very specific flavor profiles. And if we could link that to specific organisms, that would be very interesting. Eventually, it'd be fun to look at signs of domestication, especially in some of the yeast strains that we have in these cultures, because some of these mixed cultures have been used for decades within certain breweries. And if we can see that these yeast are actually exhibiting signs of domestication, that would be incredibly interesting. And finally, we want to be able to construct house cultures without problematic bugs, or maybe you can even add a desirable organism to your house mixed culture. With that, I'd like to thank GENEWIZ for helping very much with this project and giving us some fantastic reads to play with. Hank Denbacher at the University of Georgia and Andrew Brooks at Stanford for helping with the analysis. And with that, I can take any questions.
Thank you, Dr. LePage. We had a few questions come in, so we'll start off with the first one. Are there any samples you're particularly interested in?
Oh man, so one that I didn't mention too much in this presentation is actually our house lactobacillus strain that we use during our kettle souring process. We do a lot of kettle sours at Creature Comforts, and it's a technique that isn't unique to us. Other breweries do kettle sours, and we're doing that they're actually really happy if their lactobacillus is able to acidify the beer in about 24 to 48 hours. We're actually brewing a batch of kettle sour right now, and we fully expect that our house strain is going to sour that batch in about 3.5 hours. We know what species of lactobacillus it started off as. All the sequencing right now says that it's definitely not that species, but I'm really interested to take a deeper dive into its genome and see, what's special about this particular culture, that it's able to do something that no one else really sees in the industry.
Next question. What was the biggest surprise in your study?
One of the biggest surprises initially was simply how many breweries very happily said yes. These mixed cultures are things that breweries really protect, that they've taken years to build. And when we came to them and we said, hey, here's our project, would you be interested? All you have to do is send us your mixed culture. The response was phenomenal. So we're thinking we might get, 6 breweries, 10 breweries, something like that. To have 18 participating breweries was a lot of fun. Also find it really interesting to see that some of these cultures, breweries told us what they thought the samples were, but for several of them, we're seeing that they're really quite different. So samples that show markers of having spontaneous fermentation when, according to their history, they don't think that culture has ever been spontaneously fermented. And also seeing the differences between breweries. One brewery sent us 3 samples, and 2 of their samples are quite similar. You can tell that they probably came from the same brewery, and their 3rd sample is actually more closely related to a brewery that's over 1,000 miles away. So just seeing those weird differences and similarities has been a lot of fun.
Next question we have is it common for brewers to use genomic tools in their work?
Right now, as far as number of breweries, it's not very common. A lot of the larger breweries are going to be doing this kind of stuff. The 16S and ITS sequencing for mixed culture, there have been several different projects to do that. This is one of the first to use shallow shotgun metagenomics. I've seen one other project that utilized that, but that data hasn't been published. We'll also use Sanger sequencing to identify any possible contaminants. And so at Creature, we actually love GENEWIZ's service for that, and we'll send off plates every once in a while for 16S or ITS sequencing if we need to identify some organism that's floating around the brewery.
Another question. Are you planning to use insights from this data to create new mixed cultures and engineer beers with certain profiles?
Absolutely. That's one of the fun things about this project. You can think of these cultures as kind of being an ecosystem. And so if you tell a scientist just go out into an ecosystem and find all the species, it's pretty much impossible for them to know when they're done. But now that we have a really good idea of what's in these different cultures, especially our own culture, we're going to be able to isolate and separate all of the individual organisms. And with the sequencing, we will know when we're done. I will know when I have pure cultures of each of those. Our own culture, we quite like this characteristic that it has that tastes a lot like stone fruits, and we would like to accentuate that even more whenever possible. So now that we know in general what's in there, I can go through and start isolating the organisms and find which one or which mixtures of them really add to that characteristic and maybe enhance it in the future.
And our final question, would metatranscriptomics help address any outstanding questions you have?
Yeah, that would be a lot of fun. So right now we can see what bugs are present, but it'll be a little bit more difficult to figure out actually what they're doing without transcriptomics. And so I'd be particularly interested in seeing the differences between what one strain of yeast might be doing early on in fermentation and what it might be doing later in fermentation. So you might see that you have that same strain in your beer for an entire year, but by looking at its actual expression profile, you could tell, what it's doing early on versus what it's doing later. That'd be quite interesting.
Looks like in the interest of time, we'll have to stop here today. Once again, I'd like to thank Dr. Daniel LePage for his presentation and all the attendees for joining us. If you'd like to watch this presentation again, or any of the other webinars that are part of Webinar Week, the recordings will be available at genewiz.com/webinar-week.